Hello and welcome everyone. In this podcast we're going to talk about inversions and translocations. In the previous podcast we talked about duplications and deletions, and they kind of went together in many ways. Same is true with inversions and translocations. They have some similarities as well. We're going to focus on inversions first. Alright, well, let's define what an inversion is. Essentially, it's a 180 degree rearrangement of a portion of a chromosome. So let's draw a chromosome, and we'll make a centromere here. We'll have genes A and B, C, D, E, F, G, and H. And let's have our inverted sequence or region be this region here, D through F. And what happens is a double strand break occurs on the chromosome. It's cut out and then it's flipped around. And so you end up with something like this. A, B, centromere, C. I'm going to make the inversion here a different color just so we can remember it. And so instead of D, E, F, we're going to have F, E, D. Okay, so as we look at this, we might wonder why these would ever cause a problem. Right? In the non-inverted chromosome, you have genes A through H. In the inverted chromosome, you have genes A through H. You have all the genes, so why does it matter that they're in a different order? How could this cause a problem? Well, there are a few different ways this could cause a problem, and I want to mention two of those ways. The first is what I'm going to call inversions are random breaks. Where this chromosome breaks to cause the inversion is completely random. The way I drew it conveniently was on either side of the gene. But what if the break occurred here, right in the middle of gene D? Maybe it came all the way over here and in the middle of gene G. Well, we might still have a functional E and F, but the gene D has just now been cut in half. Half of it's going to remain here, the other half's over here. If you remember from transcription, there's no way you can transcribe that gene now because they're on, each half is on a separate end of the chromosome. Same with gene G. Another thing that could happen is that break maybe didn't cut the gene in half, but what if it happened right here, say, where either it left a promoter behind here, so there's no way to regulate just this gene D once we move it over here, or maybe we cut it at the terminator. We still have the whole gene, but now we cannot terminate the transcription. Those could have influences on the function of those genes. Could have very severe influences on those genes. And the second reason we might get a phenotype from these inversions is due to this term called position effect. And I went ahead and erased this picture I had before because to show this I want to draw the inversion slightly different. So let's go ahead and throw our centromere here. We'll put genes A and B here, and C, D, E, F, and G. And the inversion I'm going to have is going to occur here. Doesn't include the centromere still. And it's going to go all the way to E. And just to make life simple for us, let's just assume we have a promoter here to regulate gene C, a promoter here, and a promoter here, as such. We had spoken previously about heterochromatin and euchromatin. And we said there's a lot of heterochromatin around the centromere, as well as the ends, and sometimes in random places. And we said that genes within heterochromatin are poorly expressed. What I didn't say at that time was that genes near heterochromatin, or near the centromere, or near the telomere, are also expressed lower. It's not that they're not expressed, they just tend to have a lower level of transcription. It's the position effect of, say, gene C here. Gene C should have a lower, we would predict, gene C would have a lower expression than, say, gene D or gene E. The further you're away from these areas of heterochromatin or the centromere shown here, the further you're away, the more expression you should get from that gene. So let's draw this inversion now. And let me just draw the non-inverted parts first. And I'm going to draw the inverted part here in purple just to highlight it. And we're going to have E, D, C. And this promoter now is going to be over here, and over here. So as you can see the way I drew this now, we would have predicted that gene C in the original chromosome had a low expression and gene E had a high expression. Due to this inversion now, gene E has been placed near this chromatin area, this heterochromatin area, and now we would predict it's going to have a lower expression. And gene C has been placed in an area with higher expression because there's less heterochromatin in this area. And it could be that these genes are very precisely regulated. In fact, they probably are. Most genes are. And now we've just messed up the regulation of these genes. Gene E now is going to be much higher expressed in the original strain, and now here it's lower expressed, and vice versa with C. So these are the two reasons you might have a phenotype associated with inversion. Even though it's quite possible you're still going to have the same genetic components of these chromosomes. Alright, now I want to move into inversions and meiosis. Okay, let's talk about meiosis now. And in particular, let's talk about prophase 1. And let's draw our homologous chromosomes here. And in this first example, I'm going to show a homozygous inversion, where you have A, B, C, F, E, D, and G, H. This FED part is the inverted region, and the other homolog pair is here. A, B, C, F, E, D, G, H. Since they're the exact same, nothing has to be done for this inversion to go through meiosis. The C is going to bind the C, F, E, and D, and G, and H. It's going to be fine. Now the problem comes when we have a heterozygous inversion. So let's erase this top part and make this one here not inverted. So D, E, F. Now remember, as we're going through meiosis, the chromosomes have already gone through S phase. So there are sister chromatin here, copies of each. And because I'm trying to save room here and not make it too messy here, I'm not going to rewrite A, B, C, D, E, F, G, H on this bottom one, but you should recognize that each of these sister chromatins have the exact same genes on the other one. And same thing here with our purple example. Well, now we have a problem. This F and E and D in this order and this D, E, and F in this order cannot bind with each other. You might imagine that we could have just looped both of them out, kind of like we saw in duplications. That's not what tends to happen. What happens is, and this is always a little difficult to draw, so bear with me, we form a loop on the inverted one and then twist it. I know it sounds a little crazy here, but this is what happens. So let's draw our chromosome that hasn't been inverted yet. Something like this. And we have to draw the other one. And we'll put our centromeres here. And we might as well label these A, B, C, D, E, F. In a moment I'm going to erase this top part, I think, because it's going to start getting confusing here. And then we have G and H. Now the purple one here, what it's got to do, it's got to move in such a way where this F is going to bind here, or pair up here, synapse here, and this D is here. So what has to happen is, comes up around like so, and flips around. Same thing with its sister chromatin, of course. It's got to do the same thing because they're connected with each other. like this. So let's draw our genes on the purple. We've got A, B, we have our centromere of course, C, and now remember it's inverted so we have to go FED. And since we did this flip here, now F will be here, E will be here in the middle, and then D over here, and then G and H here. So this solves the problem, and by itself here, there won't be any problems during meiosis now. They'll synapse, they'll have crossing over. If there's crossing over outside of the loop, there won't be any problems at all, and you'll make 4 gametes. Now the problem comes when we have a crossing over event within the inverted loop region, and then the gametes we get are going to be different. And now I'm going to need to erase this top part up here because this is where I'm going to draw the gametes because I don't have any room down here. So let's erase this top part. Alright, perfect. So again, this is an inversion on the bottom. It's looped around so that they can synapse properly during prophase 1. And if there is a crossing over event, and let's go ahead and have a crossing over event, let's have it here. This looks like a good place for one. Okay, now let's go ahead and see what the meiotic products will be once they form gametes. Remember that each of these sister chromatids will be packaged up into a gamete. So this first one will be packaged into a gamete, the second one will be packaged into a gamete, the third into another gamete, and the fourth into a fourth gamete. So let's do the first one here. And as we do this, I want to use a different color, because we're going to see as we go through this, part of the chromatin is going to be left out, and I want to make sure it's easy to see that as we move forward. So the first one here, this will be one of our easier ones. So we'll do it in orange. And so we have A, B, C, D, E, F, G, and H. So we have our first gamete, which will be A, B, C, D, E, F, G, and H. Let's take this third one here, and I'm going to label it 3, just so we can keep it in order. So this is 3, and this is 4, and this will be 1 and 2. Let's do 3, because I know 3 is not going to cause too much problems. But let's see why it doesn't cause any problems. So we move along here, we pick up A, B of the purple, and C, picks up F, E, D, G, and H. Alright, perfect. So this one is going to contain, this one's going to contain our original inverted chromosome of A, B, C, F, E, D, G, H. Alright, so as long as there's no position effect or we didn't cut any of the genes in half, we should be fine. That should make a functional gamete. Now, let's go ahead and go with 2, but as you'll see, it won't really matter which one we do next, because they're going to end up at the same place. Now, let me make sure this centromere is clear here. So we're going to pick up A, B, this centromere. Now we come to this cross. Remember, now we're going to have a physical exchange of the chromosome material, so it's going to now pick up this part of the chromosome. And look what it does. It loops right back around and picks up CBA and a second centromere. So let's write this down. Remember, we picked up A, B, C, and D. This centromere. And it came down here, and now on the purple strand, it picks up E, F, C, B, A. And this second centromere. As I said before, it didn't really matter which one we started with, 2 or 4, because they all end up together because of the crossing over at this loop. And as you notice, we have 2 centromeres. So we call this a dicentric chromosome. And I'll come back to that in just a moment. I just want to finish up our map down here. And if we look, everything has been covered in orange that we've drawn through here, but we're still missing something. This purple part here has not been incorporated into any of the other chromosomes. So let's take that into consideration now. And that's going to include, and I'm going to make this a slightly different color because I want to make sure as you're going back over looking at this that you can see that this really is kind of all by itself. So this is going to be packaged up into that fourth gamete. And I'm going to put it down here and I'm going to keep it in green. And it's going to have this region here with G and H on it. Notice there is no centromere. Since it lacks a centromere, we call this an acentric chromosome. Now I want to talk about each of these four and explain whether or not they're going to help form a functional gamete. To do that, I want to erase this bottom part here because we have all the information here because these are end products of meiosis. So let's erase this bottom part. Okay, now let's talk about how these are going to form a functional gamete. And I'm going to use the same numbering system here with the same colors here just so we can remember. So this first one here, this one will form a functional gamete because it has all the genes you need in the correct order. This one that I have number 3 here will say should form a functional gamete. The reason that I say should is because we don't know for sure if that inversion has caused a position effect or if it's disrupted one of the promoters or if it cut in the middle of a gene. We don't know all that yet. But in general, this inverted chromosome should not cause a problem. Okay, this one I have number 2. I know it's a little out of order. I just want to kind of stay in line with what we have here. This creates a very interesting problem. And these dicentric chromosomes, since they have 2 chromosomes, what happens is both of them will attach to a spindle, one of those microtubules that separate out the chromosomes. And now you kind of have like this tug of war occurring in the cell, where part of the chromosome is being pulled in one direction, part is being pulled in the other direction. And this will often cause a break somewhere in the middle of here. So you might end up now with 2 chromosomes in 2, and I'm going to draw them here just so you can see what might happen. So you might end up with an A, B, C, D chromosome, missing E, F, C, B, and A, so missing a large part of that chromosome. And the other chromosome that's going to form is going to have A, B, C, F, and E. Again, missing that large part of that other chromosome, a lot of genetic material. As a result, this is going to form a, or I should say, not form gamete. Neither one of these will. Okay, let's look at this last one here, number 4. Remember, this was the acentric chromosome, and this will also not form a functional gamete. It doesn't have a centromere. Without a centromere, there's no way that it can be properly segregated during the rest of meiosis. Alright, the last thing I want to talk about is inversions and evolution. To begin this discussion, I want to draw a really brief phylogenetic tree. At this node here, we have humans, and then at this node here, we're going to have chimpanzees. So let's write that above here. Or I'm just going to write chimps. And over here, we can put a couple more in here. We can put gorillas here. And we could add additional primates as we go down. But the key point I want to make here is a few things. Just to remind you what some of these mean. Right here, this means this is the common ancestor of gorillas, chimpanzees, and humans. So they shared a certain genetic lineage at this point. And then the track that would become gorillas went this way, the track that would become humans and chimpanzees went this way. Then we have this other separation, where chimpanzees and humans had a common ancestor here. They then split, and chimpanzees went this path, and humans went this path. What's not shown on here, just to remind you, is that there are lots of different extinct species here that ultimately humans were derived from. And same thing with chimpanzees. It didn't go just from this common ancestor straight to chimpanzees. There were a lot of things in between as well. A common misconception is that humans were derived straight from a chimpanzee. That is completely false. We shared a common ancestor, is the correct way to say that. Now, during these different paths that led to humans, or that led to chimpanzees, each were faced with different natural selection mechanisms that allowed each to develop differently. Though genetically, and you've heard this before, humans and chimpanzees are strikingly similar. And I want to just throw out a few facts here. The first is that humans and chimps diverged from this common ancestor about 6 million years ago. During this time, they had a very similar genetic content between this common ancestor and the two organisms here. But what has separated them, we can look at the genetic level. And we know, by looking at the genomes between modern chimpanzees and modern humans, there have been approximately 1,500 inversions. Of which 9 have been pericentric. We'll go over that term in just a moment. Just to give an example, there's one of these inversions that occurred, and because of that, it's linked in humans to colorectal cancer. One of the big things it did, remember how we talked about those position effects and disrupting those promoters, is many genes in humans versus chimps are overexpressed. So we have a big change in expression. One key one, or several key ones, are brain transcription. Again, TXN, abbreviation for transcription factors. So in human brains, many of these transcription factors are as much as 3 times expressed. Does that explain the difference between chimps and humans? Even though at the genetic level there's not a lot of differences? It could. And some would hypothesize yes. During this time, there have been additional genetic insertions. So particularly from viruses. So I'm going to put viral insertions. So new DNA. We see differences in chimpanzees and humans because they have these different insertions that came from different species. I'm sorry, from different viruses. One such in humans is the ALU sequence that you amplified in lab this semester. Humans have that, chimpanzees don't. One such inversion involved chromosome 4. I'm going to just draw it right here. So this is the human, so I'll put H for human, and I'm going to put the centromere here. Oh, and this is also for both chimpanzees and humans. We're talking about chromosome 4. From this common ancestor to chimps and humans, there has been an inversion that has occurred. But this inversion has caused this region here to flip. Now, again, at first approximation, you might think, well, that's not going to change much. But again, remember the position effect as well as how you cut those chromosomes. Do you cut in the middle of a gene? Do you disturb its regulation somehow? 2 terms that we should mention real quick here, and then we're going to be done with inversions, is this term pericentric versus paracentric. Pericentric is an example like we see here. It includes the centromere. Paracentric does not include the centromere. The inversion does not include the centromere. And I remember this because A means without or not having. So this kind of inversion is one that does not include the centromere, paracentric. Whereas this one, pericentric with the I here, includes. Pericentric includes the centromere. Alright, that's all we have for inversions. I had originally planned to put both translocations and inversions on the same podcast, but because of some size limitations on the podcast, I'm going to cut this into 2 different ones. So I'm going to end now with the inversion podcast and give a short summary of it. And then on the next podcast, we'll pick up again with translocations. All right, let's go ahead and close with our summary of inversions. From this podcast, you should be able to draw an inversion. You should know, we didn't talk about this till towards the end, but you should know the difference between a pericentric and paracentric inversion. You should also know the special case with these inversion loops in meiosis, and some of the problems that are associated with crossing over in these inversion loops. You should also know how inversions, even though they have the same genetic content, how they can have certain phenotypes. And we talked about the position effect, and we talked about crossing over, or rather I should say chromosome breaks that lead to these inversions is random, and so that can cut genes in half and so forth. And then we closed with evolution and inversions, and how that there's evidence in chromosomes looking at different primates that suggests that inversions have been quite important in the evolution of chimpanzees and humans, just as an example. Alright, that's all I have for inversions. If you have any questions, please let me know. Thanks. Bye.